I'm Ayaka Mori-Ichioka. I belong to Osaka University and Nissin Food Holdings. I'm very happy to talk to you. I'll talk about our analysis of the interaction between hair growth key cells and other cells, the signals associated with the key cells, and the new findings on hair growth mechanisms. First, I will explain the background of our research. Hair is an important feature of mammals which performs a wide range of functions. Hair follicles consist of various components, for example, hair progenitor cells, sheath-like keratinocytes, and dermal papilla. Among them, dermal papilla DP cells are one of the most crucial elements for hair growth. The factors controlling the state of hair growth and hair loss are not fully understood. To elucidate the mechanism of hair growth centered around DP, we aim to elucidate the mechanism using two approaches. The first approach is detection of interaction between DP and other cells with healthy controls and hair loss patients. Specifically, the single-cell RNA-seq from patients with alopecia areata. Alopecia areata AA is a disease characterized by symptoms of localized hair loss. I will introduce the previous study related to the first approach. Single-cell RNA-seq in human hair follicles from healthy controls and AA patients. The author identified Wnt pathway dynamics, keratinocyte diversity, and regulatory control of hair keratinocyte differentiation. The problem of previous study is that the interaction between dermal papilla, DP, and other cells is unclear. The problem is because the cluster of DP was not detected. Therefore, we detected the subcluster of DP from the fibroblast cluster using our unique clustering approach. I will explain our methods. In step 1, we performed preprocessing, QC normalization, and scaling. In step 2, we performed integration of AA and control data with batch effect removal. In step 3, clustering and cell type annotation was performed, and in step 4, identification of DP by subclustering the cluster of fibroblasts. Finally, step 5 was cell-cell communication analysis to infer signaling pathways. We ran step 1, 3, and 4 with Sura, step 2 with Harmony, and step 5 with CellChat. The result of clustering and cell type annotation are shown. We identified 24 clusters from single-cell RNA-seq data. As a result of cell type annotation with marker genes, we assigned 10 cell types to the clusters. Subsequent subclustering results in 10 different clusters. Subcluster 8 shows high expression of marker genes for DP, and subcluster 1 shows high expression of marker genes for dermal sheath. Dermal sheath is progenitor cells of DP. The following are the result of cell-cell communication analysis. The left figure shows increased signaling in AA. The right figure shows decreased signaling in AA. The vertical axis indicates ligand-receptor pairs. The horizontal axis indicates the cell types in that pair is expressed. The closer the dot color is to red, the stronger signals. In patients with AA, the TNF signaling from myeloid and T cells to DP was increased compared to healthy control. On the other hand, the FGF7, FGFR1 signaling from DP to other fibroblasts, dermal sheath, and DP was decreased compared to healthy control. Next, I will explain the previous research methods and results of the second approach. Identification of signals released and received by DP. Alpha diversity and index of microbial diversity has been analyzed in the scalp microbiome between healthy controlled and alopecia patients. The author reported that the scalp microbiome is associated with hair loss. The problem of previous study is that no microbial strains have been identified to affect hair growth. Therefore, we screened lactic acid bacteria for hair growth promotion. We screened N793. We applied strain N793 to hair loss patients' scalps for 24 weeks to assess its hair growth effect and examined the interaction between N793 and human follicle dermal papilla cells HFDP seeds. To be specific, we performed RNA-seq co-culture with N793. In step 1, sample was prepared. In step 2, co-culture of N793 with HFDP seeds for RNA-seq was performed. In step 3, principal component analysis, hierarchical analysis, and differential expressed genes analysis with HR. The result of differential expressed genes analysis are shown. This table lists the differentially expressed genes in HFDP seeds co-cultured with N793. Compared to the control, the expression of TNFR, SF11-beta, decoy receptor, and FGF7 significantly increased in HFDP seeds co-cultured with N793. Moving on to discussion. The severity of AA is positively correlated with serum TNF level. It is also known that ruxolitinib, JAK1, JAK2 inhibitor, treated AA model DP produce FGF7. In this study, we gained 3 new insights. First, TNF-RSF14 was detected as a receptor in DP of AA patients with cell-cell communication analysis. Secondly, TNF-RSF11-beta decoy receptor was detected as a differentially expressed gene in DP co-cultured with N793. Finally, FGF7 was detected as a ligand in DP of healthy control. with cell-cell communication analysis and DP co-cultured with N793. Therefore, TNF signaling and FGF7 appears to be a crucial factor for hair growth. Last conclusion. In the halo state, TNF is produced from T cells and myeloids. TNF is received by TNF-RSF14 in expressed DP. TNF activates TNF signaling. On the other hand, in the hair growth state, TNF produced from T cell and myeloids is received by DP-expressed TNF-RSF11 beta. TNF-RSF11 beta is TNF deco receptor and is thought to inhibit TNF signaling. Since it was detected in both cell-cell communication and co-cultured RNA-seq analysis with N793, the results seem reliable. Thank you for listening.
Yeah, and thank you, Ayaka, for this very interesting talk describing a new biomolecular mechanism that associated with hair growth. Any questions from the audience? Yeah, please, Kumar.
Thank you. Very interesting finding.
Thank you.
So I worked on TNF signaling myself. So are you saying that for people who have hair loss, they have elevated level of TNF already? That's why it's causing the hair loss mechanism? That means people losing hair, TNF is important, right, in general, because it binds to the TNF receptor and affect the hair loss.
Yes.
So are you saying that people who have hair loss are generally having elevated level of TNF?
I guess hair loss patients have upregulate of TNF signaling, but other signaling is upregulate or downregulate them. I cannot understand it efficiently.
Okay, never mind. Interesting work. Thank you.
Thank you.
Yeah. Any other questions from the audience? I want to say that this...
Can I ask?
Dirk, hello?
Hi.
Please. Please.
Okay.
Yeah.
Well, I'm
Masanori, please.
Masanori from NIG, Japan. I'm interested in the interaction between follicle cells and lactobacillus. And did you try dead lactobacillus for co-culturing, or did you use only live ones, living lactobacillus?
I use medium and body. We more effect of bacterium body.
Yeah, my interest is which part of lactobacillus is affecting or interacting with hair cells. So maybe EPS extra polysaccharides or maybe some other messengers, proteins or metabolites. It's better to investigate which part of, yes.
I will study more EPS, but we didn't study EPS. I will study further.
Okay, thank you.